Okay, so hello everyone. Thank you so much for joining us today. This is the Fire Blight IPM webinar. My name is Ellie Andrews and I serve Sonoma, Marin, and Napa counties as a UC Cooperative Extension Specialty Crops Advisor. Today, we're going to hear from Jim Adaskaveg and Broc Zoller about Fire Blight Integrated Pest Management, or IPM for short. Rachel Elkins was also going to present, but she is ill today, so she's not going to be joining us, but Jim and Broc will help cover her topic, biologicals for fire blight. So with that, I'm going to hand it over to Jim.
Well, thank you very much, Ellie. I appreciate the invitation and chance to talk about fire blight. We do a lot of research on fire blight with the various Apple Commission and the Pear Board of California, and we're very excited. There's lots of interesting aspects of fire blight going on in recent years. One of the big things coming along are these, we have an SCRI grant with the USDA, and this is a multi-state grant that brings in researchers from Washington, Michigan, New York, North Carolina. And one of our goals is to look at new organic treatments or other new treatments that could help to reduce fire blight. So with that, I'm going to get started. I was asked by Ellie to cover the epidemiology of the disease, and then we'll go into some of the management practices. So I'm going to share my screen, and says I'm disabled. I have to have the host enable me to share my screen. And so this is a normal occurrence here. Okay.
Sure.
So I think you should be seeing my screen, and I can upload that. Okay, I think everybody can see the screen now, and this is the title, Epidemiology and Management of Fire Blight of Apple and Pear caused by Erwinia amylavra, North Coast region. Again, thank you, Ellie, for the invitation. Again, working with the commodity groups at both Apple and Pear in California, and this is part of my team here that works on the management practices. So fire blight's a disease of rosaceous plants. This is part of the epidemiology. As you can see, it's a bacterial disease. It can occur on all rosaceous plants, including apples and pears. The bacterium is this rod-shaped upper left corner, this rod-shaped structure with all these flagella coming out from all around the cell of the bacterial cell. And obviously, we're talking about the bacterium as a population, and so there's literally millions of these cells that cause infections on the apples and pears. The organism is called Erwinia amylavra. It's a gram-negative bacterium, and these are how we characterize the organism responsible for the disease. You can see some of the symptoms here of a flower infection, a shoot infection, and then you can see a canker. So these are aspects of how the organism can actually survive in the tree, causing infections and darkening turning the tissue very black, and it becomes necrotic, and then it wilts and dies. And the holdover cankers are the way it survives from one year to the next. And it could be a shoot canker, it could be a trunk canker, as you see on the right. And these things will ooze out the bacterium in the subsequent years, and they can survive for several years, these cankers, and they can ooze out when the conditions are warm and wet, and they'll produce the inoculum that will contaminate the new growth of the tree. So it's a pretty formidable, I think, we work on a lot of plant diseases, but fire blight is probably the most formidable pathogen we've ever worked with. It's a very aggressive pathogen, causing blossom, twig, leaf, fruit infections, limb infections, trunk, collar, and even the root stocks can be very infectious. So one of the big aspects of the organism is the production of this inoculum. And this bacterial ooze, pointed here with the blue arrow, is this droplet. And this is loaded with millions of cells of that Erwinia amylavora oozing out of that tissue. And the organism is inside there growing in between the host cells, and it releases toxins and it causes this necrotrophy, which means growing on dead tissue. And so it means that the organism likes to kill the host and then grow on the exudates that come out of the dying cells or dead cells. And that's how it survives and grows. That bacterial ooze, it dries and it's very resistant, and it can survive even up to 1 year. And sometimes under we don't see it much in California, but under really high humidity, these droplets can form strands even, and can be wind disseminated as well as splash dispersed. And even animals and insects that fly through those strands can carry the inoculum, especially if they're going to visit a flower, to the next healthy host tissue to cause the next infection. But rain does a really great job, and especially when it's warm. Broc Zoller has the Pear doctor. He puts out forecasts, and there's a lot of models, but temperature is one of the big components for forecasting the disease. So you can see the disease cycle here. The pathogen comes from these cankers, whether they're shoot or trunk cankers. They ooze out, and then the bacteria is moved by insects, wind, rain, to the flowers. The flowers then become infected, and you can see these cyclic aspects of the disease cycle where the flowers and fruit can become infected. They usually turn blackish, and later the secondary inoculum will come out of oozing out of shoot infections. And even if you get later in the season, you get fruit infections, you see these droplets coming out of the fruit. There's a pear on the left and an apple on the right in the center of the diagram here. And these things can then again be spread, and this is our secondary inoculum. So the primary inoculum comes from these overwintering cankers, and we see it causing infection, producing more inoculum. That's our secondary inoculum, and this cycle can go on. And if we don't cut out and remove the diseased tissue, they form cankers, and that's how we get from 1 year to the next. The pathogen, again, has a generation time, meaning it doubles its population in 96 minutes under the optimum conditions, which is 21 to 28 centigrade or 70 to 82 degrees Fahrenheit. The minimum temperatures, they go down to 3 to 12 centigrade to 38 to 54. So these, I call them refrigeration temperatures, still just slow it down, but it really takes off when you get into the warmer conditions. And it grows even up at 35 to 37 centigrade, which is 95 to 99 degrees Fahrenheit. So the thermal death point, we always get that with these hot summers. Does it kill it? And if we get up to 113, it might start killing back the organism in the cankers. But those are very few days, fortunately. But that's the thermal death point. And then the pH range is usually 4 to 8, so it has a wide range, but the optimum is 6 to 7.5, which is physiological pHs that occur on these crops. So the epidemiology, one of the big things that survives in those cankers, but it can also live as what we call an epiphyte. So when it's in a canker, it's an endophyte, it's inside the tissue. And when it's on the outside, we call it an epiphyte. And so it overwinters in these cankers, oozes out in the spring, and then it's rain or wind, insect disseminated. The bacterium colonizes the stigma and nectaries of the flower, and it can reach populations of 10 to the 6. That's a million colony-forming units, or CFUs, per stigma that's in a single flower. And so it can reach very high populations, and you can see how easy it would be for an insect visiting the flower for the nectar to pick up some of these cells on the inside of that flower cup and where there's high populations. And so it can then survive as an epiphyte on the surfaces, and that can even survive for several weeks, that organism. In the inside as an endophyte, I told you it could already live from 1 year to the next. And so this could be in branches and limbs and budwood and so forth. One of the biggest things in California that makes fire blight much harder to manage than in other parts of the country is that sometimes we don't have the chilling. And this results in what we call a rat tail bloom, when the tree will continue to flower. And so this rat tail bloom, obviously, if you have more flowers coming out and the bloom isn't very succinct, like in Washington, the bloom comes and 2 to 3 weeks later, it's over and there's no more flowers. So they're more concerned with shoot infections when they get rain in the subsequent spring and summer weather, where we're continually getting flowers. And so this increases the potential for infection, increases the number of protectant applications, requires more cutting out time for the workers. And again, this is a major factor between California and pretty much the rest of the country. Even North Carolina, which is lower in latitude like us, but they have cooler conditions in the spring, so they don't see, and they get cooler conditions in the winter, so they get their chilling, so the flowers come out all at once. And so our conditions make us a little bit unique as far as susceptibility to and for managing the fire blight. Now, there are rootstock differences in tree susceptibility to fire blight. Rootstocks can girdle the stem or the trunk at the base of the tree. The suckers that come up can be infected with new shoots from fire blight. And so we were out into an orchard last year, and there was a lot of they planted the trees, the signs on a very susceptible rootstock, and the trees look like in this picture B, this started to die, and there wasn't much fire blight symptoms on the top, but the rootstocks had cankers in them. And so this is on the figure A, you can see the discolored wood as you look at that rootstock. And so the rootstocks can be very susceptible to infection, and they could even have latent or asymptomatic tissue. And you could have an infection, but it doesn't really look like an infection. That's what we mean by asymptomatic. It just kind of is so slightly depressed or slightly sunken, but it looks like normal bark. So it's hard to detect this, but fortunately, we do know from our experiences that the mauling series, the M26, the M9 are very susceptible, and there are subclones of those mauling series that are also very susceptible. More moderate resistant, the M7, some of the other B9 and B118, and Geneva, up in Geneva, New York, they're developing the Geneva series, which are supposed to be more resistant. And this is a part of this SCRI project that we have to look at more resistant rootstocks. Obviously, the signs are what most people are interested in, because that's the fruiting tissue that will produce the crop. And so there's a lot of variability. Most pear trees and varieties are very susceptible, including Asian pears or apple pears as they're known now. Shinko, I actually grow Shinko in my farm, so I can do research on it without the tree dying, for example, but it is a little bit less susceptible, is how I would categorize it. But Bartlett is very susceptible. Bosc, red Bartlett, and this star crimson are really very susceptible. Where I think moderately susceptible would be Danjou, Secco, and Comise. And the Comise, we actually can keep the orchard alive, but it is susceptible, but it doesn't outright die like a Bartlett if you leave it unmanaged. Apple, again, very wide-ranging susceptibility between the signs, different fruiting woods, and Granny Smith gets the disease. But it seems to be a little bit less severe, where the tree doesn't necessarily get structural damage to the scaffold branches. The cankers don't run, but it does get disease from these the flower infections and the shoot infections. Obviously, more susceptible would be Golden Delicious, Granny Smith, Galveston, Jonathan, and so forth. These are all susceptible, and highly susceptible. I listed as Gala and Fuji, that if you don't manage it, it becomes a very prone to dying. So we see that managing fire blight is going to take a lot of efforts. The organism is a very tough bacterium, and it knows how to survive, and it knows how to spread itself. So we have to start right with our planning of an orchard, with our rootstock sign selection. That's one of the key. People overlook that. Like I was telling you, last summer we went out to a field and they overlooked the rootstock that they were growing, and that's like the first step that you would want to do in planning a new orchard. Obviously, selecting more resistant rootstocks would give you a better chance of managing the disease. The orchard location, and some locations higher up are cooler, like if we go up to the lake in Mendocino County areas, it's cooler there. So it's not, the pathogen isn't going to be under its optimal conditions. But when you get down into the Delta in Sacramento County, it's the climate is different there, and we get a lot more air moving, but yet we get a lot more warmer temperatures. And so again, there's differences even within the state of California between how intense the fire blight situation can develop. As far as managing, you want to avoid the high nitrogen fertilization. You got to fertilize the trees, but you don't want to put excess nitrogen, and avoiding over irrigation, because if you have a lot of water, this is how sprinklers that get up into the canopy, this would be, again, a very dangerous situation with the warm temperatures of inducing the splashing and the dissemination of the pathogen, which will result in more disease. Sanitation, as you get fire blight, you have to think about removing that tissue. That's the pruning aspect of cutting the cankers before they get into the main scaffold branches, and destroying them. And that means burning them or burying them, but it's a lot of work to bury them. But in the past, we tried to destroy them by burning them. In season, again, removing the strikes before they run down. Usually you have to go 12 to 16 inches beyond where the tissue's dying. So because bacteria moved in the vascular tissue, and they can be found. And we did a study with the Pear board a few years ago showing that you have to go at least 12 inches, but most people say 12 to 16 to make sure they get that shoot removed before it gets into the main scaffold branch. Removal of alternate hosts. We know the urbanization is taking place. People grow all kinds of rosaceous plants, Cotoneaster, Crataegus, Cydonia, and Pyracantha are some of the more common rosaceous ornamentals that are also very susceptible. So if you're near urban areas, then these plants can be hosts, and therefore they could be windblown, disseminated their inoculum to your orchard. And then, when you're cutting, you have to clean your tools. The bacterium is this very small bacterial cell, probably no more than 1 to 2 microns in size. And so this means that these bacteria can contaminate in their slime, in that droplet that came out, which is full of plant host sugars and as well as the bacterium. And these things get like spread over your equipment. And if you don't sanitize, you could, when you make a cut, you could actually be introducing that bacterial slime onto the healthy tissue. So historically, we've used treatments, chemical treatments to manage fire blight, starting in the early days with copper. And we moved to antibiotics more recent in the more recent time, but now we're under a lot of regulatory scrutiny about some of these materials, and we're moving into some more fancier things like growth regulators, like Apogee, and there's other generic materials, and SARs, which are systemic acquired resistant treatments that stimulate the host to become resistant. That's like Actigard. And I'll talk a little bit about this later in the talk here, but and then rotation between the different modes of action. So, we've been very lucky. We had streptomycin. People say, oh, it developed resistance, but it was introduced back in the 1950s. And Oxytec came along in the late 60s, early 70s. And, then fortunately we got kasugamycin registered just in 2018 in California, and as only the third antibiotic registered in U.S. agricultural history. And so this antibiotic kasugamycin is not immune to resistance. It has to be used in rotation with other products. And so we'll talk a little bit more about that as well. So we do have antibiotics. We're fortunate that to manage this disease, we have those, but we're also looking at early season copper treatments to reduce the inoculum, but we don't want to use them too late in the season because of the potential of phytotoxicity and the potential of developing resistance that's kind of a response to the copper applications. So we do screening of fire blight isolates, the Erwinia amylavera strains. We test them to the antibiotics, but we also look at them from their sensitivity to copper. You can see that they seem to grow. These are strains on a nutrient agar plate, and we put them in a radial fashion so we can see the growth response. When we put 20 parts per million in the agar, you can see the doesn't really affect growth. It's a little bit weaker, but when we get to 30 parts per million, you can see that the colonies are starting to separate. They're not all merged together. And if we use a poor medium, even at 15 part per million, you can see differential aspects of the strains. Some strains can still grow, but at 15 part per million, but other strains have a harder time. And that broken up thing, what the arrow is showing is the colonies are separating and not able to grow as a homogeneous strand. So we envision that the environment that we're using it in the early season, there's not much susceptible tissue, which means it's kind of a poor medium to grow. And so the copper would have an effect even at 15 or 20 parts per million to reduce the bacterial population without causing phyto. So this is like a pre-bloom and bloom applications of copper would be helpful to prevent the inoculum from building up. What we're showing here on the right here is that over time, if I leave these plates, they'll eventually, these bacteria won't just, they're sitting there kind of stagnant. But if you see how they grow after 2 weeks on that same medium, the copper doesn't disappear, the bacteria will start to grow. And again, this is what we call an adaptation to the copper material. So we basically are looking at nutrient poor agar as a representing the early season when there's not much nutrients out there. The buds are just starting to push. And again, when the flowers come out, it's just an early aspect of the season. We can get reduced growth with 15 to 20 part per million metallic copper equivalent, or MCE. But if we continue to use copper, we observe the spontaneous mutations forming, and we get the production of these cells will respond, and they will grow in the presence of the copper. So fortunately, there's no high resistance. We see that we can grow these bacteria even up to 50 part per million as an adaptation to the copper. But initially, they can be reduced their growth even 10 to 15 part per million. And so this is more of a spontaneous mutations that occur. And I think the best way to describe it is an adaptation to a more harsh environment. Bacteria respond, so they don't turn on all their genes all at once and are running everything in their library of DNA that they have. So what they do is they have constitutive enzymes that are being to keep the cell alive, but then they have enzymes that are responding to the environment. So if you keep using copper, it'll turn on some aspects of its DNA that will give it resistance to the copper by chelating the copper and then allowing the organism to grow. So copper is rated in the IPM efficacy tables as a plus or a plus plus. It's mainly being recommended as a contact material. It's not being something that's going to have a long residual in the sense of with reach back or penetration into the host. It suppresses the growth of the pathogen, but in the long term, the bacteria can respond by turning on these adaptive enzymes and resume growth. We have looked at copper, the rates, 0.5 of COSI 3000, which is about 33% metallic copper equivalent, is about 180 parts per million if you use a half a pound per acre. And this concentration, again, is diluted when you spray it on the plant, and it gets down to around 18 to 25 parts per million, which, again, is the concentration that should be effective in those early sprays under poor nutrient conditions. Under highly favorable environments, you can get moderate copper resistance isolates that can grow above 25 part per million, and you can get spontaneous mutations occurring from these adaptive enzymes that are occurring. So if you're going to use copper, we want you to use it in the early season during the bloom period, but later sprays might not be as effective, but they can also cause some russeting of fruit. We do screening of in vitro sensitivity of Erwinia amylavra to antibiotics. In this case, we're using a spiral plate assay, and this is where the concentration of the antibiotic is the highest in the middle of the plate and lowest at the edge of the plate. And so you can see here, we plated out moderate resistant ones, which are the ones that are going halfway across the plate, highly sensitive isolate, which means it's only growing at the very lowest concentration of the antibiotic, which is to the far left right here. And we can see that we have even resistant isolates that grow across the entire plate. So in this case, we have the concentrations is that at the center here is greater than 70 parts per million, or milligrams per liter, is considered highly resistant. Moderate resistant is where we see this growth stopping right around here, and that's about 20 parts per million on the plate. And then we have sensitive, which is when it goes back here, and this concentration is 0.6. So we basically get a 2.5 log concentration gradient when we do these spiral plates, and we can learn a lot about the sensitivity of these bacteria to the toxicant, in this case, streptomycin. So we've done a lot of work with the Pear board and identifying the different types of resistance. We have this strep A and strep B, which gives us moderate resistance, and they're associated with this transposon TN5393, and we can generate these different types of, we can observe, I should say, these different types of resistance levels from sensitive to moderate resistant to the high resistant. One of the ways we summarize this in a paper we did in collaboration with George Sundin in Michigan was that we have this chromosomal resistance in California, Oregon, where there's high resistance. It's only been found limited places in Michigan. We also find that there's this moderate resistance, which is these things that are still sensitive to strep, but they're inhibited at the higher concentrations. And this is from this strep A and B genes. And these can be present in different plasmids. And we had initially the PEA 8.7 was the strep A and B resistance gene. But then we also found this new plasmid, this PEU 30, which is similar to what's going on with these other plasmids that are found in Michigan and New York, where these plasmids have also convey this strep A and B resistance in this transposon. Well, what that means is that we could monitor these different antibiotics for resistance. And so we've done this over from 2021, 22, 23. I just give you a feel how we're looking at the antibiotics. You can see in 23, we had moderate and high resistant strains being detected in our Sacramento County. Lake County, they were all sensitive, and Yolo County was moderate resistant. So the moderate resistant is that strep A and B, where the high resistance is that chromosome resistance, and that we can still detect that. So we have the different types of resistance to streptomycin. In 23, we had all the isolates that we received were susceptible to oxytetracycline and kasugamycin. In 22, we had some moderate resistance detected for oxytet. In 21, we had high resistance of oxytetracycline detected. And this is one of the first reports ever of high resistance to oxytetracycline. And you can see on the left here that we've been doing these surveys for over 15 years to provide information about the levels of resistance to strep, oxytet. And now we've been monitoring kasugamycin, but we haven't detected resistance to date, even though it's only been introduced since 2018 to 2024. So that's we're going in our 6th year of using that material. Again, the levels of resistance are, when you have high resistance, it's 100 parts per million. We have moderate resistance, we just say it's 4 part per million. And when sensitive, it's usually just some fraction of a part per million. So unfortunately, we have to be aware of this, both apple and pear growers. If we overuse oxytetracycline, we can select for resistance. I think that's the take-home message. And we already know that with streptomycin. And we want to get that impression to growers that we can't overuse kasugamycin either, because it will be, these organisms are really going to be adaptive and come up with solutions to how to survive. And one way is to develop resistance. And so we're afraid that if we overuse kasugamycin, we'll get resistance. So our strategy is to interuse or rotate between these materials to keep ahead of the bacterium for selecting for resistance. And as just a little update on the oxytet, again, we characterize this, and we're finding very high levels of the Erwinia amylava resistant. You can see we had sensitive isolates and resistant isolates, and that was what we call the moderate resistant. And then we found these high resistant isolates that grow across the entire Petri dish. We characterize these, and just to show that there is not an anomaly. They are pathogenic, and what we're showing here is that the oxysensitive can be reduced by treating with oxytetracycline. We can significantly reduce the number of infections with these are just single applications. So we went from 50% down to 22% or so, a significant reduction in the incidence of fire blight with one oxytetracycline application. When we characterized our oxytet resistance, it was also resistant to streptomycin. And you can see a single application was statistically similar to the untreated control. So resistant isolates are not inhibited by 100 part per million. They're highly resistant to strep, so they have both. They have dual resistance. They're not controlled by a single application, as I just demonstrated with labeled rates of oxytetracycline or streptomycin, which are at 100 and 200 parts per million. They're competitive with wild types, so they cause disease, so they can compete against the wild type. And we're learning about their persistence, and we think that in the absence of oxytet, they go down. But these plasmids are hard to get removed from the population. So this is what a plasmid is here. We characterized that, and it was published in this paper here. But it's a circular piece of DNA that carries here's the strep A and B resistant genes, and then it has this tet B, which is also the resistance for the oxytetracycline. So there is a plasmid. This is the first time it was ever described, this PX11-7, that has both the strep and the tetracycline resistance. So our goal is to evaluate, ways to prevent selection for resistance, looking at other materials and using kasugamycin in combination with other antimicrobials, including copper materials. You're looking at new formulations of oxytetracycline that have higher concentration, and again, using those in mixtures or rotation so we don't select for that plasmid in that resistant strain. Using copper sulfate in these newer coppers that are based on copper soap like Cueva, or copper sulfate pentahydrate, which is Mastercop, or CS 2005 are some examples of those. We're identifying new materials. We have these food preservatives approved by the FDA. They're synthesized materials by biofermentation, and it's called epsilon polylysine or EPL and niacin. And these are materials that are additive to bakery goods and other things, to other food items approved by the FDA. So we're looking at these types of food preservatives as potential new treatments to help manage fire blight. And there are other things we're looking at. This up in Oregon, they're using aluminum potassium sulfate, or alum, ALUM, as a treatment. This isn't registered yet, but there are companies trying to move towards registration of this material for fire blight management. So we wanted to evaluate that. Cinnamon aldehyde, SACAM, and cinnamon oils are cinerate, and we're testing these materials as well. So most of these materials here are organically certified, and we're trying to integrate them with the antibiotic materials to try to preserve their usage while still getting high efficacy. So we've looked at a lot of materials. This is just a summary here, the antibiotics that are registered in California and the US. We're looking at natural products. These are the FRAC codes. This is the Fungicide Resistance Action Committee. You can see right away that these antibiotics have different modes of action. So this is why we're rotating to not select for any one type of resistance to this mode or that mode. And you can see right away that this kasugamycin is different from the others, and it's different from the others not only in their mode of action, but also in the fact that kasugamycin is used only in plant agriculture. It's not used in human or animal medicine. So then we're looking at natural products, and the FRAC code system uses BM, or biological material 01, for anything that belongs to plant extracts or natural products. Fermentation products would be in that. And so you can see that we're testing things like DART, which is acid, caprylic and capric capric acid. This acacia bark extract is QAM. Cinnamon aldehyde is SACAM. And cinerate is a cinnamon oil. We're looking at thyme oil, thyme guard, and this alum, which is potassium aluminum sulfate. The EPA is reviewing this, and we hope to get it registered, but we wanted to get some efficacy data. And then we have some numbered experimental compounds like this bacterial metabolite RAA that also has some promise. The food preservatives, I mentioned niacin and epsilon polylysine. And here, as I mentioned, we're mixing it with EPL here to see how well it can perform. And that's called JAX1. There are things like Blossom Protect, which is this yeast. This is a fungal organism trying to manage the bacterial organism by colonizing the flower ahead of the bacterium. So one of the big things in microbiology is site exclusion. If you're first to the table, you're able to protect that flower from infection. So other companies now are trying their yeast. YSY is a yeast. And we know that other companies use other bacteria to fight the bacteria. And this is like Serenade. They have fermentation of Bacillus subtilis. And then we're looking at ways to mix things to protect the antibiotics. And so we're looking, these are ongoing materials for evaluation. As you can see, one of the common things here with these food additives like polylysine and niacin, they're polymers. And you can see this is also a polymer, and it's repeated. And so this structure is, again, presumed to interact with the bacterial's membrane. And we think that other products like capric and caprylic acid, which have this same type of chain, binds to the membranes of these bacteria and prevents them from functioning properly. So interestingly, dodine, which is commonly known as a fungicide, sold as Sillet, is also like a polymer-like structure. And again, we think these things are interfering with the bacterial membrane's function. And so that's why we're pursuing those. We have a little laboratory assay that we do on ornamental pears. We do this because we get these flowers in January, and they're flowering down in Southern California, and we can inoculate them. You can see the fire blight running down the pedicels of these flowers, causing the black and characteristic fire blight. And you can see we tested our JAX1, which is the cinnamon aldehyde mixed with EPL, and you don't see hardly any black pedicels. And then you can see the Cassumin was very effective as well. That's our standard. We're comparing it to new materials. So we see that there's a rate effect here, and JAX48 is working really well, comparable to Cassumin. And again, a lot of the treatments were effective, significantly reducing disease, but we're looking for something that would be commercially acceptable. So we want to get down into this area here. This is 70% disease in the control, and we're looking at down to 5 to 12, 13% for the JAX. So we're pretty happy with that performance. So in 2022, we did some work in live oak, looking at new bactericides. And you can see that here's our QAM material. That's this acacia species. We have cinerate, which is registered, and it's doing fairly well in all our trials from 21 to 22 and 23 at 32 fluid ounces. It's doing quite well. This is the 60% material, and I know that they have a new formulation out, and so we're trying to figure out how that is going to work. But this data is with the 60% cinerate formulation. And we have mixtures of Cassumin with the EPL. These are low rates of Cassumin mixed with the KFD is a UPL formulation of EPL. And we're mixing that with the zinc material and getting fairly good results with the EPL and the niacin materials. And then I'm pointing out here that Cassumin significantly reduced the disease, but we're mixing it with Sillet or mixing it with Firewall, we can get really high performance there. And this is, again, we're getting very high levels of control from 30% or so down to just fractions of percents, 1 to 2% of the disease. And then in 23, the Jax material was evaluated here at 48 and 32 ounces, significantly reducing it from the controls. The Cinerate, again, performing very consistently in our trials. This is, again, the Jax will be an organic. The Cinerate is an organic approved material. And we're trying the Quam as well as the RAA materials, both A and B, looking at their performance. And again, mixing Cassumin with Sillet gave us the highest level of control. But this RAA mixed with a surfactant like Regulate gave very similar results. And our Jax 1 at 32 fluid ounces did really well. So this is a mixture of SACAM with the EPL. So NUP is the new oxytetracycline formulation from New Farm. And this is just a higher concentration of the oxytet so that we could use only 8 ounces instead of 16 ounces, and we can get very good disease control. And the Quam did really well, again, mixed with DART. So we are making progress to find effective natural products and trying to mix these things in with our integrated program. We can get effective disease control. So we did a study, part of the SCRI grant that we had in this multi-state trial was using the same treatments. On pears, the alum didn't do well. We had multiple years of data showing that it didn't do very well on pears. The Cinerate didn't perform well, but this is a really high level of disease. Again, 30% of the shoots on the tree were infected from flower infections. Jax at 32 did okay, but it wasn't spectacular, brought it down to about 12%. Oxidate, similar, around 10%. Even Firewall was around 8 or 9%. And then the Blossom Protect we were really happy with. It worked really well. And Thyme Guard did pretty well in this trial. So then we turn ourselves to some of our apple trials. This is Fire Blight on Granny Smith apples down at the Kearney Agricultural Center in Fresno County. And again, we had the apples aren't as severely infected, but we did have about 10.5% here with the control. The Jax did pretty good at 32, but look at 48 ounces, it's less than 3%. The Cassumin mixed with Sillet are again right around 3%. And then the surprise here was the alum doing really well. And this is what most of the other people in that SCRI grant, which are doing the work on apples, say alum's working really good. And so they were surprised that when I told them it wasn't working very good on pears, but it does work really good on apples. And then I show them this data and they go, well, that's like we're getting. So we can reproduce this effect across the country on apples, but it's really hard to get that effect on pears. The Blossom Protect again did really well, and the Cinerate did really well in these trials. So we're moving ahead. We hope to continue to try these materials. We're looking at, again, the new formulations of the antibiotics. We're looking at mixtures of the antibiotics with some of these materials like Dodine or the Maniplex zinc. We're looking at these combinations like Jax1 of these food preservatives with cinnamon aldehyde. And then we're looking at cinnamon oil, which is the Cinerate, the SACAM, the QAM as potential standard treatments that could be developed to put into rotation, reduces the antibiotic use while still giving us high performance. So I summarized this into these circle charts because the circle charts kind of give you an idea of the modes of action. And these are based on the FRAC codes. So we have copper, manzate, Sillet. And then we have the antibiotics. You can see they're all different FRAC codes. And then we have other materials that the phosphonates we've been evaluating. They change the pH and they suppress the infection, but they're not really standalone. So we want to use these in mixtures. Actigard is being used by my fellow SCRI grant people in Michigan and New York and Washington, mixed with Apogee. They like a two to one ratio, 2 ounces of the Actigard with 1 ounce of the Apogee, and they call it the 2-to-1 mixture, and they get really growth suppression of the new shoots, which again for them is a big infection site because they get a lot of rain back east, and they don't want these new shoots from getting infected. And so this combination of Actigard mixed with Apogee, 1 to 2 applications at the beginning of shoot development seems to suppress that, and they get less fire blight. Again, part of the SCRI is to look at blossom protect, Cinerate, and these other materials, and look at them. And then we're still looking at new materials like another yeast, very similar to blossom protect, colonizing the flower ahead of the fire blight pathogen. QAM, RAA, and JAX1 would be suppressing the bacterial population, so they're antagonists to the Erwinia. So these kinds of things give us a view of what our treatment choices are or will be with these experimentals and how we can integrate these for planning our management program. So we have a summary here. There's differences among the cultivars for pears, Bartlett, Camise, Winternellis. But with apples, I mentioned that some can tolerate it without getting cankers, but some can be devastated, and we know that. Rootstocks can be an important selection process for preventing really lots of fire blight, and so you want to have things that can not have a lot of succulent growth, but yet control the tree's height and give you tree vigor. So you're still looking for rootstocks that are more resistant and not going to make the sign more susceptible either. So that's part of the planning, like what varieties you want to grow and what rootstocks you choose. Cultural practices, I mentioned before, you want to make sure you manage your nitrogen, avoid over irrigation, and then again, orchard location and design allowing for more air movement would be better. How do you do that? Well, orientation of the orchard with the prevailing wind. So the rows going with north-south are more likely to have more air movement than with prevailing winds coming from the north or northwest. Then, we're not getting the movement if we have an east-west trellis system. Pruning out is very important. Removal of infected tissues and removal of alternate hosts. That's all part of the sanitation practice. Chemical control, obviously, I spent a lot of time on toxicants like copper, antibiotics, and natural products. We are looking at these non-antibiotic bactericides, things that antagonize or could be under biocontrols, antibiosis, antagonization, parasitism of the host, which would be Erwinia in this case. And so we're looking at these as well. Using systemic acquired resistance, Actigard, and then using Apogee as a growth regulator. These might be combination treatments. Insect control, again, very important. Okay, so this is all summarized in these things we monitor for populations, and then this is summarized in the UCIPM website. This is found under UCIPM agricultural pests, and on the bottom of the page it says it has this document here, which gives you the apple and pear bactericide conventional as well as the biocontrols and natural products with some rating systems to show you and help you design a program. Well, thank you very much for your attention. I know I went a little bit long here, but Rachel wasn't available to talk about some of the biocontrol, so I covered some of that for her. And I think you got a feel for what's available and what we're trying to accomplish in the near future.
Thank you so much, Jim. I really appreciate it and appreciate your flexibility covering the biologicals as well, since Rachel's ill today. Yeah, I think it looks like we have a couple of questions in the Q&A. Are you able to access that? Would you prefer me to read them off?
Yeah, just read them off. I can exit out, but maybe they want me to return to a slide or something.
Yeah, that's a good point. Okay, so the first one is from Kamyar. So he's asking, what is the entry point or point court for rootstock trunk infection? So, and I remember I saw this Q&A pop up when you showed us that rootstock that got infected and how folks maybe just kind of missed that since it's lower down on the tree. So, what is the entry point for rootstock trunk infections?
Well, in many cases, it's these what we I don't like to use the word suckers, but they're new shoots that come out of the rootstock, and these things are very susceptible when they're new, and so you can get in wet conditions. If you even have micro sprinklers hitting that young shoot, this is an infection, and then it grows down from that apical tip. It grows down the water sprout is maybe a better term than sucker, but it goes down into the base of that rootstock, and then it can cause a canker, and that's the usual way. If you get an injury there, or if you have a lot of water there, and the bacteria is oozing out from other infections, it gets splashed dispersed, it can infect if it's overwatered as well. So the bacterium can get into that tissue, but it likes the real delicate new tissue, and that's why we see shoot infections, flower infections. These are very delicate tissues, and that's the main mechanism for getting into those rootstocks.
Thanks. Okay. And then he also asked, if ornamental alternate hosts are present but asymptomatic, can they be considered clean, or are they a substantial source of the pathogen even without any symptoms?
Well, I don't have a lot of experience with studying this, but this is in the literature. Herb Aldwinkle in New York published on this. There's papers on these asymptomatic alternate hosts, if you will. And, if all the rosaceous plants are considered possible hosts, I have a hard time thinking about an asymptomatic host carrying the pathogen not being a source of inoculum. I mean, it has to be causing some disease on that alternate host. And if it is, then it would be oozing it out, but it might not be very obvious. But, with insects moving these droplets of ooze coming out, and they might not be as prolific producing these ooze droplets as we might see on a pear or an apple. But I think that it would be a source of inoculum at a certain time, and maybe it oozes 10% less or 50% less. But if it's raining when it's oozing, it can be a source of inoculum. But again, it's not going to show a typical symptom like a dying canker or a dying branch. It might just form a little suppressed canker, but there is enough to produce inoculum when it gets rained on and wet. So again, I don't think these symptomless ones are going to be overwhelming inoculum sources, but if you're working so hard to keep your orchard clean, and you have some of these alternate hosts there that potentially can provide inoculum, you might be shooting yourself in the foot by having them in close proximity to your orchard.
Thanks. Yeah, that all makes a lot of sense. Our next question is from Michael, and he's asking any data or comments regarding potassium phosphate.
Well, potassium phosphate, we've tried it. It does change the pH. It's more acidic. Bacteria generally like this more neutral, optimal condition. So the phosphate is completely systemic, and it moves through up and down the plant from a foliar application or a soil application. It'll go into the plant, and it does move. It's totally what we call ambimobile, meaning it goes from the top down or from the bottom up. And so it moves and it acidifies, and so it's going to be suppressing the growth of the bacterium. And in our trials, it helped to reduce it, but I think if you used it in combination with other bactericides, you might see some benefits from that. We haven't done extensive work on it, but when we used the product by itself, it was more, I would characterize it as more of a suppression than the an actual like a management for control like an antibiotic. So if you're trying to get levels of disease management that is, up in this where your control's at 30% and you want less than 2 or 3%, that's pretty hard to do with phosphite. But if you put it on and you get down to 15% or 18%, that's what I mean by suppression. So it helps to reduce the disease by slowing the organism down, but it's not going to give you a function as a standalone and give you high performance in reducing the disease. So I think that's the way you should look at the phosphites.
Great. Okay. Next question is from Pat Carlson asking, just to double check, did you say that folks are using a 2-to-1 ratio of Actigard to Apogee in their sprays?
Yes, that's 2 ounces of the Actigard and 1 ounce of the Apogee, and they're applying it 1 to 2 times, again, depending on their vigor of their trees and the environmental conditions. This is taken across multiple palm fruit growing areas, especially apple growing areas in the US. So that's the ratio that they're using with 1 to 2 applications to help reduce that shoot growth.
Okay.
It's usually applied after bloom, as the new shoots start to grow. Back east, they say they put it on in late April and early May. But again, that's all dependent on the variety and the bloom stage and so forth. But we'd have to kind of think about that in terms of our shoot growth timing. So that could be maybe as early as April, first 2 or 3 weeks of April. So we would have to adjust for California conditions versus what they're talking about for Eastern and other parts of the United States.
Okay, we have a series of questions from Vince Trotter next. So we're going to start with first one. Does fire blight kill buds along a branch or trunk? We've seen extensive blind wood in some varieties and wonder if that's attributable to fire blight. And then the next one is, can you talk about the threshold between manageable and game over? But we can start with that first one for now.
Well, that's a difficult question. Bud death, it depends on the time of year that you're seeing the bud death. Pseudomonas syringae is the bacterial blast pathogen. It affects multiple crops, including apples and pears. We can see bud death from that organism. So it is complicated to say so the time of year would have to be kind of put into there. Usually you see this bud death in the early spring or late winter from cold conditions, and then that would be more leading towards this other bacterium, Pseudomonas syringae, Pathovar syringae, which is a well-known pathogen of killing buds of tree crops. The second part of that question, I didn't really understand. What was that again?
Oh, sure. So can you talk a little bit more about the threshold between manageable and game over? So where is that threshold, between where it's manageable and you can work with it versus maybe it's more worth it to remove the tree? And he follows up with the next question saying, with cankers on a trunk, for instance, is there nothing good to come from keeping the tree? So just sort of at what point does it make sense to remove that tree?
I don't have a lot of experience with that, but maybe Broc can help me answer that. When you get into the trunks, it's really hard to you start hacking the tree down it's so much, it's hard to see you're going to get fruiting wood being produced. You know, on the station, once we see trunk infections, it's pretty hard to keep a tree going. Now, for pear, but if we had an apple, you know, maybe we can keep that alive. And usually the spreading of the bacterium inside the tree of an apple is a lot less, it's a lot slower and it's a lot less extensive than what we see in pears, for example. I think fire blight in Bartlett pear is the hardest thing I've ever seen to try to control. It's once it gets into the trunks, and it's not salvageable. But that's my take on it. I would ask some of the PCAs that have direct experience with this, and our next speaker will probably be able to answer that better.
Thanks. Yeah, so I'm thinking in the interest of time, since we're a little bit over halfway through the 2 hours, it probably makes sense to move on to Broc's presentation. But Jin, if you're willing, it would be great if you wouldn't mind typing your answer to the questions we haven't addressed yet in the Q&A section. And then I think everyone should be able to see that in the chat. And we can also come back to some of them after Broc's presentation. And maybe Broc, if you're willing to share some thoughts on those questions too. So I think we'll move on. And Broc, are you able to share your screen?
Like it's on there, is it there?
Yes.
Yes, looks great.
Okay, maybe I could talk about a couple of questions while we're thinking about them. So one of the questions was, when do you decide to push an orchard out? Well, I think you probably have to look at what percent of the trunks in your orchard have it. It's very difficult to manage at that point, and you're just going to have to decide, and it's going to be based on price of what you're growing, if I think it's worthwhile to survive, those kinds of things. So, but basically, if it's in the trunk, it's going to be very difficult, really, very difficult. And the other question was talking about the phosphites. I have seen that used, and it seemed to me that it does stop the tree from growing along with the blight. And I think it has some effect on fruit size also because of that. But I think if blight is so bad that you think you have to consider that, the thing to do is put it on just immediately when you find out you're in a lot of trouble. So anyway, we can talk about that a little bit when we get a little farther into my presentation, I guess. Anyway, I'm going to spend some time talking about the degree hour system for managing fire blight treatments, and then spend some time talking about new worries about blight, which are some of the things that Jim was researching there. And I've been on the research committee for the Pear Advisory Board for probably 30 years, and I can tell you we never have turned down a fire blight project that anybody submitted. It's that important a disease to the pear industry, and I don't think we ever will. And so it's just extremely important to us, and we're constantly having to rotate tools. And even today, we're so glad we have Kasugamycin, even though it took about 6 years longer for us to get it to use here in California. We're glad we have it. On the other hand, we're only able to use it a couple of times a year, and we can't use it after the main bloom is over. And so it's not really substituting for a whole lot of what we've lost, really, with resistance to the other antibiotics, but we're very glad we have it. Anyway, moving along here, let's see if I can okay. Let's see, going back here. I went too far, so if I can think about how to go back here.
You can use the arrows on your keyboard if you'd like. That's
the
that's
right.
way I
That's
do
right.
it anyway.
Yes, you told me. Okay. Okay, so why do you need a management system for fire blight anyway? Why don't you just go out there and put the treatments on? Well, a big reason that we face, in some of our pear country is, we've got the place messed up already in a year like this when it's wet. Trying to get on treatments early for psylla, things like that, trying to get on scab treatments. And so, I tell the grower, you got to get in there, it's full bloom now. You got to get in there and start spray for blight because we've had these heat units. But, if we don't have the heat units, maybe we don't have to get in there again. So That's one of the reasons you don't want to have to be in there all the time spraying for a disease, even as important as it is. So here we are at full bloom plus a day in one of the years, and these were ruts that were put in there earlier, and you can imagine how hard it is. I had some photos, which unfortunately I lost, of equipment being stuck over the years, and it's not that hard to get them stuck. Anyway, in the early 70s, we did there was a medium that was developed by Terry Miller and Milton Schroth down at UC Berkeley for culturing fire blight bacteria, and this was really a magical thing. And I can tell you that when I first got into plant pathology, I was totally interested in fungi, really, because you could look at them through microscopes and see what you had, and it was just totally interesting. Well, unfortunately, I got drafted out of graduate school, and I ended up. I thought they were going to send me to Vietnam, but they sent me back to the Biological Warfare Center back in Maryland. I spent 2 years back there culturing bacteria, and I got a little more interested in them. And so when I got back out of there, Miller and Schroth had just developed this medium when I was finishing up on a PhD, and it was something I was quite interested in. And there was a company up in Yuba City in the mid-Sacramento Delta area there, which is one of the most susceptible areas in California for blight. And we grow pears in California because it doesn't get blight as bad as back on the East Coast. That's why they don't have a Mediterranean climate there. It rains in the summertime, and we don't have that here. So at least there's kind of an end for our blight season in the year after spring. So that's why we have pears here, but that doesn't mean it's that way every year. So the upper Sacramento district was always just a little bit warmer. It was away from the opening out to the bay. And so it was warmer in the winter, and the chilling wasn't quite as good. And so the pear bloom was not quite as crisp. It took a longer period of time. In the Lake County coastal mountain area, it was the crispest that it is, because it's very cold up there in the winter. So the dormancy is very good up there. And when they decide to bloom, it comes out a little more rapidly. So we don't have as long a bloom period to deal with it. And it's also a little colder. So that part of it is nice. But the worst part was the upper Sacramento district. And that's where this company was headquartered that I went to work for. And they wanted to, they had clients there and also in the Delta. And Rachel Carson had just come out with her book Silent Spring, and the pear industry was very interested in reducing pesticide use in their crop. And so there were grants available for growers to participate in this. And so they submitted samples to the company there, Agricultural Advisors in Yuba City. And they were already in the business of growing virgin female codling moths to put in traps so that the males would come to the traps attracted to the females, and that's how they could monitor codling moth. This was before the development of pheromone techniques commercially. And this is kind of the same thing, really. This was a brand new thing that was developed in the lab there at UC Berkeley. And we went out and we did sampling there, and it showed a high correlation with the number of degree hours that accumulated above 65 degrees Fahrenheit prior to sampling collection. And that was very important. If you take a look at the the analysis, the R-squared values here, 0.9 for each of these particular areas, Sacramento County and Lake County, you can see it's very high. It's pretty difficult to get that kind of correlation with field studies, I've found. But we looked at for 5 years in these areas, Sacramento Valley and Lake County, and we looked at the percent of the samples. And these are the numbers of samples that we took in each area. And there were, in the course of 5 years, there were about 2,000 Sacramento Valley samples, and there were, I think, about 300 or 400 of Lake County samples. And a sample was 100 bacteria, 100 blossoms that were picked randomly and put into sacks and brought to the lab area. And we plated out about a tenth of a milliliter or so on the selected medium, and then we put them into incubator and grew them until the bacteria could be seen as colonies on the plates. And there was a pretty close relative to Erwinia amylava that grew on the medium. It was called Erwinia orbicola. And you could tell the difference looking at them. But just to be sure, we actually took samples of every single one of these colony types and put and introduced it into green pear fruit and put them also in the incubator in a glass of water and looked to see that the bacteria actually were the pathogens. So it took a lot of work to do this. And of course, the 5 years, why it was pretty certain really that you could monitor them using degree hours. And this 65 was not an old concept really. Mills back in New York years before that in the 50s had decided that if it rained and there'd never been any 65-degree weather, there wasn't going to be any blight in the apples and pears they had. And so it was just very logical to insert 65 into this. So we tried some other temperatures too, but it really worked really quite well with 65. So one of the things that showed up here in the Sacramento Valley, which is the dark units here, it's stuck on 0 here, and about when it left 100-degree hours in the next sample period, which had been 200, we started picking up the bacteria. Whereas in Lake County, it was a little bit later. They were there in the 0 period up to 200, and so we use 150 as a threshold value in the Sacramento Valley and 250 in Lake County because of this. We've used it ever since. Well, as an example, here I am cutting some blight here a few years ago in 2015, and this is on a Seckel pear, and I don't do anything to my home fruit. And it's something of a test really for our area to see how blight is doing. And of course, we're looking at the weather and we're doing degree hour sampling and so forth. But it's always good to have trees that see that aren't being managed, how things are doing. And so Seckel trees actually are somewhat resistant to blight. They get the blight and a lot of the blossoms fall off without the blight running in. A lot of the ones that run in a little farther stop before they get to the trunks. And it actually is not absolutely necessary to cut it all out, but it was so bad this particular year. It was virtually every cluster, it seemed like, on the tree. And it was one of these seasons where we had over 600-degree hours above 65 and it had rained. And so one of the things my growers taught me years ago is you never want to cut blight out of your own tree. You're not going to cut far enough down into the next trunk to avoid the disease. And they're absolutely right. On the other hand, if I'd waited any longer and cut every branch, I wouldn't have any tree left. So I decided I was going to get out there immediately as soon as I saw it and get as much as I could off the ground. And so there's a couple hundred on the ground right there. And you'll see the last slide in the presentation. There was more that came later, but the tree is still there. It survived this. Now, one of the things that we've seen is an infection period is when you've had degree hours a certain level and you've had rain or high humidity at certain temperatures. And it takes a while for the disease really to become visible. So the first thing that becomes visible if you're really looking is this bead of ooze here. And this particular flower here is a little bit black and where it came down. And this particular event was 1 to 2 weeks ahead of when you first ahead of this particular period. So what I found is when you see something like this, you're looking at an infection period that occurred 1 to 2 weeks earlier, and it depends on how warm it's been since that particular period. Sometimes, it might be 6 days, but usually it's about 10, 14, that kind of thing. And of course, you really have to be hunting for it. You're not going to see this bead of ooze there on this healthy looking tissue. You're not going to see it for a couple, 3 weeks later when it moves farther down and starts blackening and flagging things. So that's one of the issues that you deal with here. It occurs, and it's occurred a long time before you ever get to see it. One of the issues that we deal with here in California, of course, as was mentioned, is that we don't just have a main bloom like they do in apples. We also have a lot of secondary bloom. So here's some on shoot tips, which is the last covered up on my screen, but it's the last ones that appear. There are many more that occur between main bloom and this, however, that you have to deal with. This one is a little bit easier to handle because a lot of times it'll get to be above 100 by then and it'll stop running, and oftentimes you don't even have to cut them. But in between then, there's some that come directly out of the wood, pruning cuts, and those go right into the wood, and they have to be dealt with very, very aggressively. Try to prevent them. This is the shoot blight here, which it's kind of the same concept. Here's a bead of ooze. You're not going to see it immediately. This one I saw because it was starting to turn a little bit black, but you don't know whether this was weather related or whether an insect actually, got a hold of some fire blight and moved it around. But the end result is the same. This was some flowering pear on Kelsey in Kelseyville downtown, and these were planted quite a while ago. But I would say probably 20, 30 years ago, maybe, but nobody ever remembered this happening. But in January 2015, they virtually all got blight, totally, just 100% across all of them. And they're blooming right now, and the pears are just starting to be a blossom or two out there. So they bloom ahead of Bartlett's. And we don't normally have a lot of degree hours accumulating ahead of the Bartlett bloom, but in this particular year, there was a number of them accumulated, and then it rained, and it was ahead of the Bartlett bloom. It didn't really affect the Bartlett bloom till later. It got some of them, but this particular flowering pear is still an ornamental in downtown Kelseyville along the streets. They managed to cut it out, and it's there still, but this hadn't happened to them before. Let's see here. I got to go back. No, wrong way. Okay, we talked about, monitoring in 1972 to 1976. Well, in 76 to 85, we started counting infection numbers to get an idea of what the effect, how many new infections we would be getting based on degree hours. And we're talking about degree hours plus an infection period that we have here from the first blossom stage through full bloom plus 15 days. So that's the area of time in the calendar when most of the blossoms are there. It's not nearly the end of blossom season. It's another month to go after that, but they're not there. And those are actually occurring during periods when there's a lot of degree hours and the rains are very effective on them. You have to keep the treatments going. But if you should make the mistake and not treat for a situation that occurred during first blossom through 15 days, you're going to have a terrible situation, just unbelievable, one that doesn't happen every year. So it's a period that we sort of base our action on, really, as far as being ultimately aggressive. So here was random blight per holdover, and I'm looking at blight out there in the orchard in these counts that not really next to holdovers. I'm counting holdovers, but if there's blight next to them, I'm not counting it because it probably got there from rain splash. So what I'm interested in random blight is stuff that's out in the orchard where I'm not really visually seeing the holdovers and that got carried by the insects. When I first got into this, seeing was really believing, because I worked in the upper Sacramento Valley there, and a lot of people had pollinizers there and beehives, and they got better crops actually by using them, but actually Bartlett pears don't require them if the temperature is good enough. They make a pear without seeds in it. And in the entire Sacramento Delta area, there are no cross-pollination used, but in the upper valley there was. And anyway, when I first went to work out there, I parked myself by a beehive, and there was essentially nothing coming out of the beehive till it got to be 56, 57 degrees. And then you got up past 60, 65, 70, all of a sudden the sky is just covered with them. And of course, they're out there visiting all the flowers. And so it's just seeing is believing, really. The farther you're away from the holdover and the warmer it is, the more the bees are getting it around, and other insects as well. Their temperature requirements are quite similar. So anyway, there was a pretty good straight line here. Again, some pretty high statistical relationship. So at this point, we decided, okay, so this is predicting that we're going to get a worse blight situation when we get up in these high degree hours that's occurred with an infection period, first blossom, full bloom. Why don't we try to do something about that? What can we do? We started the thing up, following what we do with the average temperature system, where it predicts that the bacteria are there. And so we'd start going every 5 days. But this is what we got from every 5 days. The more bacteria, the more the degree hours occurred, the more blight we were going to get. So we decided, well, let's try narrowing the treatment gap when we get into some higher degree hours. And so in 1986 was the first year we tried that, and it turned out to be a year that we had 600 degree hours in the upper valley, and so that was a tough situation. We went every other day with treatments during that period, and we got down here, which it would have taken having no degree hours at all to have gotten that before. So that was a sign it was working, and we followed that. So this graph is kind of the same thing. It goes up through 2002. And so these are what we just talked about here. This is 72 to 72 to 76 is when we were just looking at the bacteria. And we also continued to look at degree hours for forever after that. We still do it. And this is the blight per holdover that we counted in the middle Sacramento area. And this was the 1986 time when we first decided we would increase the number of treatments. And all of a sudden we weren't meshing with the number of bacteria with the heat units. I'd also like to point out that this heat units is random new blight times 100 per holdover. And the reason I put it up to 100 because it happened to match the ups and downs of the heat units themselves. Now, I to be honest with you, I can't do that anymore. When you see a little later in here, we're getting so much blight now, I have to only multiply by 10. Otherwise, I lose some of the blight data. It's so high. But we continued this from 86 until 2002, and by and large, we were getting a lot less blight. It wasn't perfect. There were times we made mistakes. This was one particular time when my grower had a mushroom. He ate the wrong mushroom and had to go to the hospital and couldn't get the extra treatments on. So that's when we also got a lot of blight. But the next year, we were back into it and everything was fine. Now I would like to talk about holdovers a little bit here. So you could look at holdovers, and you could look at random strikes per holdover, and you could look at random strikes, per degree hour. So if you're looking at per holdover, why it's a lot higher statistical situation here. So the R-squared value is much higher than if you just look at random strikes here. So the number, and this is what the R-squared value is. So it's much more statistically valid to be looking at both. And the reason is the holdovers are just incredibly important source of bacteria. So the more of them you have out there, the more new infections you're going to have. So this just readdresses the importance of holdover removal really in fighting this disease. If you don't do it, it's like adding another 40 blight strikes. Each holdover you don't remove. Did it again. So let's just address the degree hour system again. 1 degree hour is 1 degree above 65 for an hour. So if you had 70 degrees for 2 hours, that would generate 5. That would generate 10 degree hours more. So you're looking at every hour of the day that's above 65 and adding degrees hours for that particular day and adding them all together. And you accumulate them every day. Until you get 3 consecutive days. If you do below 66, in this case, you would reduce the accumulated value to 0 and start all over again. The exception is if you've had the accumulated degree hour total and you haven't had it reduced by cool temperatures and you got rained on, or the humidity was in coincidence with 57 degrees or above, you don't reduce the degree hour total. And the reason is you may have got a whole lot of new infections at that point and you won't be able to see them for another month. And so you have to not take chance that those bacteria are gone. As a matter of fact, if you went out and did your sampling in the blossoms, you wouldn't find them there. But what if you got a whole lot of new infections in every tree from this last rainfall because the degree hour total had gotten up above 400 and you hadn't seen them yet? That means if it rained at that point, you're going to have rain splash infections all around those. So you have to be very careful about this. And that's why that's in there. You don't reduce the total if it's gone through one of these heavy degree hour infection periods. It's only if it stayed below it. Okay, so there's a couple thresholds we use as we discussed, and it's because the bacteria show up a little later above 0 in Lake County, but in Sacramento Valley, they start showing up after 150. So if you're below 1 degree hour, you don't have to do anything. And this would be the case that Mills was talking about back in New York. He didn't see any problem with that. And so we've always used it because we never picked up any bacteria anyway. But at 150, we started picking them up. And so we've used 1 to 150 as for spraying in the 24 hours prior to a predicted rain. And we consider 1 to 150 is still in that 0 area. And that's why we say it's okay to wait until you get a predicted rain, because we think if you don't get the spray on, the end result is not going to be terrible. So, but it's still a good idea. It should be done. But if you don't get it on, you may get away with it just fine. So, but you get to the next threshold, 150 to 500, all of a sudden the bacteria out there, they show up in samples, and you don't want to miss having on a treatment ahead of a rain or predicted humid, warm weather. So you're out there on an interval at that point, and it's every 3 to 4 days at that point. You get above 500, and you're still in the into 0 to 15 days past full bloom period with real heavy bloom. Or sometimes we've treated extra later when for some reason there's a heavier bloom than we think we should be dealing with. Sometimes we'll maintain the heavy treatments a little longer than that. But basically, get over 500, treat every other day during the major bloom period up through 15 days. At some point, you decide to quit, and hopefully it's when you get a little bit higher temperatures that the bloom starts to fall off. So then you can decide, okay, I can't quit yet, but I'll increase the treatment intervals back to 4 or 5 days, or even 6 sometimes, and everything will be fine. And so in California, in the Upper Valley, why there's typically there'll be some 95 to 100 degree weather, probably about the same time you're running out of bloom. In the Sacramento Delta area, unfortunately, some years they never quite run out of bloom, and so it's a little bit harder to decide when to quit. Usually they start quitting because we want to put some water on, because we're the first district to pick and we want to have some fruit size here by the time harvest comes. So a lot of times they'll start the water a little earlier down there in the Delta for that reason. But later districts, we typically will not start the water until we don't have blossoms anymore. And it's very helpful to do that. Now the treatments are half treatments that are applied every other row. They're reported as one treatment. So if you spray every other day, you're reporting that you did a treatment every 4 days, which is what you're doing. You're treating the whole orchard there every 4 days. But if you're doing every other day, it's putting on the half treatment every other row, and you're allowing more blossoms to open, because if you're into one of these heavy degree hour situations, you're getting more blossoms opening in those situations than you're when you don't have any. And so those blossoms, they have a tendency to get reinfected by or recolonized by the insects that are flying around when the warm weather is there, less so when it's cold. And so that's one of the reasons that we have to keep going there and keep the treatments close together. Now in the coastal mountain area, we're adding 100 degree hours to each one of these units. So we're not doing anything up until we get to 150 here in the coastal mountain area. And when we get to 250, and we get to 250, we'll start doing the regular treatments. And we actually have not done the every other row very often up there, but there's some years we wish we had. And so we're doing a little more often, and we'll talk a little bit about that a little bit later. Now, this used to be available online, and I don't think it is anymore, but this is kind of a graphic of what went on in 2018 in Kelseyville. And the reality of it is that all it is something to talk about. This doesn't really help you, because let's say you're right at this point on the 31st of March. You don't know anything at all about what's coming here. All of that is just unknown. All you know is what has happened. So what you're most interested in is getting reports of what's predicted to be occurring in the next 10 days. And there's incredible data that you can get online, really. Doesn't work out perfectly all the time, but it's extremely helpful. You can see what's going on in the next 10 days and what you should be doing. So it could tell you that it's going to stay warm, or there's going to be a front coming through. Sometimes the rainless fronts are the ones that are scariest, because they raise the high dew temperature at night. So there's something you have to be looking for. So basically, if the degree hours continue, you have to continue. But if it says, and you haven't gone over the 400 or 500 threshold, that it's going to drop down and do nothing, why then you figure, well, I'm not going to do anything for a while till it starts over. So anyway, it's useful to talk about later, because it has the degree hours in there. This is the degree hours for this particular season for this district. This is the rainfall. This is the average temperature, if you want to look at that line. It has amounts of rainfall, which is another thing. There's something that could be added to the system, in that we've seen that there can be too much rain sometimes. So we can get high degree hour levels, and I'm still not entirely sure which amount of rain to use, but a couple of years ago we had 0.75 hundredths of an inch of rain for a storm. Another time before that we had an inch or more. I think there's been 3 times when I've seen extremely heavy rain after a lot of degree hours that should have been a good blight infection period, but the rain was heavy enough, instead of washing the bacteria from the stigma onto the nectary, so I had to wash them off on the ground. So there wasn't any infection. So it's magical, but it does happen. And I don't tell people to not be spraying for situations like that. They should be putting the treatments on anyway, but if it happens, it's a gift. Okay, if we look at growth here, this is, let's see, I'm losing the pop up there. Not sure which that is. Not sure what district that is looking at it. Can anybody read the top? It's one of our districts anyway. So this is
Kelseyville.
This is the Kelseyville district. Okay. All right. So this is the bloom period that meshes with an infection period from early bloom, the first blossoms, to 15 degrees after full bloom, that has meshed with an infection period. So this is the highest infection period level that's occurred in each one of those particular year's seasons. So you can see, to get these high levels, it doesn't happen every year. And but it does seem to be increasing a little bit. It's not horribly statistically significant, but it does seem to be increasing. I would say it's more likely that it's cyclic. That's what you're liable to see, which is another thing to talk about when you have these cyclic downturns here. This is almost generational. Here this looks like a 6 or 8, 10-year period when there's not a whole lot of blight weather. And so you have some new people working on your farm. Maybe your son's coming in and managing things, and he's never experienced the bad situations that develop here. And so his attitude maybe would be different about blight than the one you have or should have. So this, like I say, you have to pay attention to what the degree days are telling you, not so much what occurred in past years, because it just changes immediately in 1 year. So you have to be ready to change your attitude. This particular one, I think, is Walnut Grove, and we're seeing the most change down there, and I'm not exactly sure why. One of the reasons I know is that the bloom, I believe, is just slightly later and it's more strung out. And it's because they don't have the fog in the winter they used to down there. That's one thing I remember going to school at UC Davis, that, I'd drive home to where my folks lived in Nevada, and it just was totally fog bound driving up and down the Central Valley. And that was a normal situation for maybe 2 or 3 months really in the year. And we don't see that so much lately. So we're not getting the chilling, and the blossoms are not as crisp. And so, and the bloom in general, when you reach full bloom, is a little bit later in the year, it looks like, along with the degree hours that you've accumulated and the infection periods. So that's something to consider. There was extremely bad blight during these particular years. And this one in particular was bad because it had been a number of years really since anybody had any blights. This was really bad. And this happens really. It's a cyclic thing. Here's a period when there wasn't a whole lot. Here's a couple of years when there wasn't. So it can change in 1 year. And this is the mid-Sacramento Valley. I can't quite get as far out on that one because, there essentially are no orchards there anymore. Blight was part of it. The economics of pear raising it wasn't as good. And those are the orchards that went out first. And you can see there, up there, their bloom time was also becoming warmer, although it was also a cyclic thing, but we don't have the last few years, which were upward bound to look at. But if you put these all in the same graph together, this was always the worst district as far as heat. Okay, here's the same district, Upper Sac again. And so here's the data that I showed you earlier, where I was multiplying blight for holdover times 100. And it made this particular set of data mesh against degree hours perfectly. But look what's happening now. Look at all the blight that they were getting here. If this one's covered up by the screen, but it's way up here. If I wanted to put that on the same screen, I'd have to and I multiplied blight by 100, it'd be way up here somewhere. So this is how much more blight really was occurring in the Upper Sacramento district, which is the place where the resistance developed first, really, to some of the antibiotics, along with the Walnut Grove area, which wasn't as cold as wasn't as warm as the Upper Valley, so it's a little bit tougher. Anyway, a little bit slower, but it's developed there also. So you can see there was, if you looked at the trend line, blight was going up. If you look at the heat of the temperatures of the infection periods, it was going up also in this district, and they're not there anymore. So these are changes to consider. One of them is a reduction in the chilling hours in the dormant season, and so that means the bloom is going to be extended. It's going to be a little later, and it's extended over a longer period of time. And so that actually is going to affect blight potential. Increasing resistance to oxytetracycline. Fortunately, I guess it wasn't found last year in samples, but it was the year before, I think. And so it's something to consider. This particular material lasted a lot longer than streptomycin did. But I think it's showing some decline also, and we had to take some pressure off of it. Greater contribution from other hosts of Erwinia amylavera. They planted more cherries down in the Sacramento Delta. Of course, there are almonds and everything everywhere now. And so there are direct neighbors to pears, which are alternate hosts for the bacteria, at least, even though they don't show symptoms. So your pear trees, they could be getting the bees or insects that could be visiting cankers over there in your pear orchard. And then they fly back over to the cherries and whatever to get the pollen and nectar over there, because they're blooming already. They bloom before the pears. So what we have seen is that where you do have neighbors like this, or if you've got a home that's got one of these things next to it that's an alternate host, flowering plum or something like that, very often you're going to have a little more blight right in that particular corner. So it's something to think about. And I think it's increasing just when the temperature is increasing and the insects being as motile as they are. So it's made us, interested in doing other things, really. And so an old treatment really is just treating with high levels of copper prior to bloom. And it worked with apples quite a bit, and it was used in apples. The apples have a much crisper bloom, and they don't have rat tails. And so if you could delay the bacteria from leaving the cankers, I guess, and not getting to the bloom, why, it made some sense. And we, in fact, did try it in pears, although we discounted it because we thought, well, the pear bloom is so long, this is not going to do us any good. But there were times when we tried it, when we had so much holdover out there, we thought, we got to do everything we can. And we were pretty sure it actually did something. We had a number of blocks where we did that, and the neighbor didn't, and the neighbor had quite a bit more blight. And so we thought that it did work. When we started getting more resistance to antibiotics, Rachel got interested, thank goodness, in doing a research project on it with Ken Johnson, and I helped a little. But anyway, the samples were taken and they were sent up to Ken's lab, and he measured the bacteria levels in the samples, since we weren't doing that anymore. And he found that in 3 out of 4 years, he got at least a 2-week delay, sometimes even a 3-week delay, it looked like, in the bacteria that were showing up in blossoms. So this could get you through, could get you at least partially through main bloom, maybe even farther. So anyway, it's something we're doing for the last 8 years. We've been doing this now. We've been putting it on with delayed oil, which actually helps psylla control. And so we're out there spraying anyway. And so we mix for the second half of it, as long as the green tissue isn't out yet. We put some high levels of copper in there. You can't put it on green tissue because these levels will probably be injurious. But it looks like it works, and it's something that we've added to the situation. Jim talked about his trials, but this year he presented this one also in the book, and I think he talked about it there. But things like cinerate and cinnamaldehyde, they look pretty good, really, and essentially we're almost as good as some of the antibiotics we had here. Which, like I say, we love Cassumin, but it really doesn't add a big part of the season to us because of the rules we have to follow. And it actually, putting it on later, that can cause fruit finish problem. I never really got registered for using later than that. So anyway, those are issues we deal with. The biologicals, that was always somewhat of an issue too, because we couldn't use our fungicides, when we had biologicals out there, if we were in scab areas, especially, which we are in the North Coast. So how do you really mesh those together? It's kind of tough, but the Cinerate and the Cycan, those have some fungicidal properties as well as insecticidal. So we're kind of interested in those, and we're going to try to put them in. We thought we'd do it last year on a small scale, and we didn't get it done, but we're going to try it again. And we need to get some experience with them, looking at them, what they do to fruit finish, what we can mix with them. Can we put on iron treatments, for example, with one of these materials and not destroy fruit finish? There's just a lot of things we have to know. So, as well as if we wanted to mix them with other fungicides, some experience needs to be done. But we're very interested in what's being done there. So anyway, this was a couple of weeks after I got done the first time cutting, and these are ones that I missed up there. So you can see the cuts are a little bit bigger, but the same thing. There probably some I'm leaving there in the tree, and as a matter of fact, there was some that ran in a little farther. But basically, I'm allowed to do this because these sickles, it doesn't run very far and very fast in them. So I would advise that to people that are homeowners. The sickles are a tiny little pear, extremely tasty, but they reject the blight. This one is an Asian pear, Shinseki, which it's unbelievable how fast blight travels in Shinseki, but they tend to bloom a little bit early, which in Lake County, most of the time anyway, they miss it. But when they don't, it's terrible. And they also, they're serving them as a source for bacteria from other trees there. So anyway, those are problems you have to deal with if you're not treating for blight, and it's extremely difficult, really. Pears are a extremely difficult crop to grow. Anyway, if anybody got any questions, I guess we could deal with them. Let's see.
Thank you so much, Brock. Would you like me to read them to you, or do you want to look at them?
Let me see if I can get back on the screen here. New share. So let's see. This question's up here, I guess. Well, what should I answer here?
Yeah, great question. So I think, we left off partway through Vince's question, and I think maybe we could just pick up there where, he's saying he's heard sanitation of pruners isn't really such an issue during the dormant season and before bud break, and he's wondering if that is true.
I have read that, and I believe that's true. I think it's extremely important if you're doing it, if you're pruning late, but and if you're cutting blight, of course, but during the true growing season, I think it doesn't really matter. So.
Okay, thanks. I had actually asked that same question to Rachel like 2 months ago, and she said the same thing. Great question. Okay, also the next one is, how long does copper retain efficacy on the tree? Is application during dormant season of any use?
Well, we try to put it on as late as we can without there being any green tissue. And so that's always something you have to worry about. And I think it probably lasts longer if you don't get any rain between then and full bloom, but you're probably going to, or early bloom. And so it's going to affect the life cycle of it. And maybe that's why it only worked 3 years out of 4. I'm not sure. But I think there's a limit to it. There's no doubt. But it seems to be worth doing from what we've seen and what the research says. And so, like I say, we're already putting on an oil spray. And so it was just very easy to add the copper to it on an every other row basis.
Okay, great. And I see Jim is typing an answer to Vince's last question. So for now, we'll go to Zia's question. So Zia Sonabend asked, are there any of the biologicals like thyme oil or Cinerate that have been used after bloom is completely over? We have very late infections getting into our quince.
Well, those are things we have to find out, really. I don't know a thing about, what the Cinerate does or the thyme oil to fruit finish and things like that. And so we have to get experience on that, because every year is a little bit different. And even if you had good luck when you did the research trial, it doesn't necessarily mean the next year when you try that it's going to be the same. So it's kind of like the Blossom Protect yeast. You know, there's some years when it doesn't do nearly as much harm, but the harm it does when it does occur, it makes it, we can't use it in Bartlett pears. We could use it in Bosc, but even that one we kind of worry about. It makes the Bosc crack a little bit. So much russet. So unfortunately, that's one that we can't use, but it's like I say, you have to find out by trying them. And we start out on a small scale, you know, maybe a few acres, a few tanks or whatever, and try it for a while and just see.
I have a quick question actually related to that. I had heard that Blossom Protect, when you use it in combination with Buffer Protect, which adjusts the pH, it could have higher efficacy. Have you seen that out here in California at all, or not yet?
I think that's the way that they sell it. I think the two are mixed together, and which, like I say, as soon as we saw that we are going to have trouble with russet, we haven't got any more experience with it. But there were a couple years we used it, and
Yeah, the russeting seems like it could be a big
you
problem.
know,
Okay. Thanks for letting me insert that real quick. So another question from Vince, we're wondering, when applying chemical controls, should we be applying to the orchard floor as well?
I don't think that matters. I'm not sure it would do it. It might do something if you got stuff, you know, that you've cut off. That is a problem. There's some counties where you can't burn anymore. So if you think you should be burning it, there's some counties where you can't legally do that. They probably won't be growing pears much longer, is the way I look at it.
Catherine P is asking, where can I buy small quantities of suggested antibiotics or other treatments for a home orchard? She says she has a Gravenstein, very infected, and Golden Delicious, no apparent infection, as well as one each of Fluot Prune, Santa Rosa Plum, and persimmon trees. So where could folks who have just really small orchards look to buy small amounts?
I think you can buy Cinerate and Cycan probably online, I think, in small quantities, but I'm not so sure about the antibiotics. I think you'd have to go down to a commercial place that sells to growers, and you would have to have a permit. It would have to be reported to the ag commissioner, that kind of thing. So that's a problem really with a lot of the things that you have to use on your home orchard, really. There used to be a lot of things you could try, but they're just not there in the stores anymore. So what do you do on a home orchard? I don't know.
Well, quick question, going back to removing disease material, since, you know, for folks who can't or don't want to burn diseased pruning material, do you have suggestion for how else to dispose of it? I know that comes up a lot, at least with folks I've been talking to.
Well, Jim said bury them. That's a lot of work. And the hole would be awfully big in some cases. So like I say, we can still burn in Lake County, and they can in Sacramento County, but you can't in San Joaquin County.
Yeah, yeah. A couple more questions here.
Yeah.
We've got Michael is asking or saying that I think this would have been towards the tail end of Jim's answering questions, that you mentioned a pretty low rate of apogee, and Michael's asking if there's a synergistic effect. So this ship might have sailed for that question. I can't quite remember the context, but Michael, if you want to type in more details, feel free.
I see that in there. I haven't tried a lot of apogee, so I don't have an opinion on that.
Okay. Camiar is asking, what are considerations for managing fire blight on loquats, which bloom in late fall?
So I'm trying to think. Loquats, of course, get it. I think they bloom a little ahead of pears, but I can't remember, to be honest with you. My grandmother raised them. I don't know, but it's the same issue. What really can you use? I think one of these that you can legally buy would be the ones to try, really. Like I say, I think you can get a hold of Cinerate and Cycan. I think there's some organic materials that you can buy small quantities of too also.
Yeah, and I do appreciate what you mentioned too about trying things out, you know, on a small scale first, seeing how things go, maybe doing some comparisons, and then, you know, evaluating that first before you decide to scale up. Another question, we have 1 or 2 minutes left. Scott O asked, is apogee advisable on pears?
Well, I guess it's had some data to show that it does some good, I guess. I think it's one of those things that you would use on young orchards myself, is what I think about it. But I haven't paid attention to that one because it hasn't added a lot, and so it hasn't been on my list of things to consider, but it may be in the future.
Okay,
But we have tried, like I say, we have tried phosphite, and well, most of the time when I saw it applied, it was applied, after they saw they had a whole lot of blight, and they got very close to the time when the first 95 to 100-degree weather occurred. And so the strikes ended up stopping anyway. So it was that definitely was too late to put it on because they were going to stop anyway. But I think if you got into one of these heavy degree hour situations and it rained on and you hadn't done anything and you got a lot of holdovers out there, maybe you'd want to consider putting on some phosphate and just immediately, maybe even before you saw anything, but at least look and see if you start to see something, put it on immediately, because then at least you'll get a couple of months out of it. Otherwise, if you wait longer than that, you're not really going to get a whole lot out of it that Mother Nature's not doing for you anyway.
Okay, we'll do one or two more questions. So Vince asked, are there resources out there that track accumulated degree hours the way CIMMYS tracks accumulated rainfall? And I know there is something called the Cougar Blight model out of Washington State, but I'd love to hear from you two, what you think about that model, if there are other ones.
Well, you used to be able to go to UCIPM and they had the model on there, and they still cite it as something to use, the degree hour model. They cite all the models, but there used to be a program there that you could key into that would give you the degree hours, same as it gives you degree days for codling moth and things like that. I went in there the other night, and I don't think it's hooked up anymore, to be honest with you. But the reality of it is you get that, and it is nice to have the degree hours, but I have weather stations and I go track them myself. That's what I do. And we have a local facility in Lake County Weather System. And so they give you the highs and lows, and you can get it hourly, and you just write it down in your logbook or keep track of it. So that's the way I do it. But it used to be you could go to UCIPM, and if you didn't want to track it yourself, it would be tracked for you as of a certain date.
So for folks who are maybe close to CIMMYS stations, that might be a good resource just to track things, unless they have their own.
CIMMYS are good stations. The thing about them is they don't submit the data as soon as other stations, and so you have to wait almost a day sometimes to get the answer, which is later than you want if you have to deal with this disease.
Yeah, that's a great point. Okay. And last one, okay, maybe two more real quick if you guys have time. Can you describe proper sanitation of pruning equipment? And then, oh, it sounds like Tanya Lee is saying, I'm checking why the model isn't turned on. Okay, so one last question. Can you describe proper sanitation of pruning equipment?
Well, bleach works, but like I say, I don't think it's a practical solution for pruning in the wintertime, because so many cuts are made. Your pruners are not going to be putting bleach on every time they make a cut. Nothing would ever get done. And according to people's experiences and whatever data there is, there's no need for it anyway. But you do need to worry about it if you're cutting blight, or if you're pruning late enough in a warm season. You should be worrying about that. And so there are some things like bleach works. Just spray bleach on them.
Okay, well, with that, I think we'll wrap up. Any last comments, Brock or Jim?
No last comments. All the questions were answered really well, I thought, and we'll have to get back on, some of that mixture of the Apogee and the Actigard. I don't have direct experience with that mixture. I was reporting from the SCRI grant researchers who are using it back east. So I will look into that and see if I can get an answer for that.
Thanks, that sounds great. Well, thank you both so much for your expertise in this area. I really appreciate it. I know I learned a ton, and I just put a link to a survey in the chat. So everyone, if you don't mind just taking 1 minute, it's really short, I promise, to fill out this evaluation survey. That just gives me feedback about this webinar. And I really appreciate your input. There's also a little section for comments if you want to write out any comments as well. So with that, I think we'll wrap things up. Thank you both so much. And I'll be posting this on my YouTube channel soon for anyone who wasn't here. Everyone's welcome to feel free to share it with folks as well.